And good morning, Advanced Kentucky AP Biology students and teachers. Welcome to our fourth of the content review sessions to help you review and get ready for the AP Biology exam on May 18th. As we get started, please do turn off your microphone and your video, but feel free to use a chat box. If you'll take a minute now to tell us where you're from, that will be terrific. We will be monitoring the chat box for questions. But please don't use your microphone. Our speaker today is MaryBeth Cook, and many of you may remember her from a few weeks ago. MaryBeth teaches at Harrison County High School and is a reader for AP Biology, and so she will have some sage advice for you. I'm Lou Akampora, the Science Director for Advanced Kentucky, and I'll be manning the call-in show, I guess. Again, please turn off your microphone and video, but do use the chat box. This session and others are archived at kyap2020.com, along with some support materials for all the sessions. So please feel free to look there for any help that you might want. So again, here's MaryBeth, and her topic today is the cell cycle and regulation. So I will turn this over to her. All right, MaryBeth, you're on, and feel free to share your screen.
Okay, does that look okay?
It's a little slow right now.
Okay.
Oh, there you go. It just came up for me. So you're on.
All right, guys, welcome back. And today we are going to talk about cell communication. I'm hoping that we'll actually get into the cell cycle, but I'm not sure that we'll make it there. So Lou, when he posts this video, there will be a PDF of my slides, and then there are some notes that I've typed up so that you can look at that and kind of review the material that way. Okay, so what I'd like us to start, I want you guys to go to chat and go ahead and answer these two questions, just to get us going, see what you guys are thinking this morning. So what I've tried to do is give you a lot of images in this presentation and try to tie in all the stuff that we have discussed in the past and that you've learned in those different sessions with Brittany and Robert. And my suggestion to you is, when you see an image that you don't know, try to orient yourself and kind of pick out the stuff that you do know and seems familiar to you. My chat always goes away. There we go. All right, so I'm getting some good answers. So most of you are saying this is a plant cell, good job. And the openings or the pores, I'm getting some different answers here, but I did see plasmodesmata. So that's what I was wanting you to go with there. And so once you know this is a plant cell, that's going to help you answer some questions that they may throw your way. So as far as this, as far as cell communication goes, there's three main ways that cell communication can occur. And this first picture is showing you direct contact. So these two cells are touching directly, and you can still see with the arrows that they are sharing information. Okay, so another example is immune cells. So the immune system's been removed from our curriculum, so you may or may not have gone over this in your class, but this is also just another great picture of two cells directly touching and sharing information. So you have the host cell is labeled here, and then you have a T cell that's come in the picture. Again, you have this perfect little shape that's allowing these two cells to talk, and the T cell can help take care of the pathogen. Okay, so that was direct contact, two cells directly touching to share information. The next way that cells can communicate is through short distances. So they would be close to one another, and molecules can usually diffuse from one cell to another to share their information or have those cells to act. So Lou is going to set up a poll for us. And what you're seeing here are two nerve cells. And let me see. And again, knowing the details of nerve cells is not something that you have to know. So they would provide the information that you need to know. But what I really like about this picture is not only can we tie in cell communication, but we can tie in all those different types of transport that you discussed this year. And so what would happen is this cell, this top cell in this picture, sodium ions will be rushing in. So that's how the message is traveling down that neuron. When all that positive charge builds up at the bottom, that's going to be a signal for calcium ions to move in. So my first question for you is, how is this calcium ion making its way into the cell? So what type of transport has to occur to get in? So some of you are already voting.
So most of us have voted. I'm going to give you about 5 more seconds to make your choice, and then we'll see how the consensus is. And 3, 2, okay.
All right, good. So most of us did say facilitated diffusion. If you're looking at this picture, and I know it is kind of hard to tell, it seems to blend in there. There is a transport protein there. And so calcium is charged. So charged or very large molecules have to go through a protein to get through that phospholipid bilayer. So good job on that one. When calcium ions enter, that's going to kind of trigger the next step. And so that's going to be a signal for these synaptic vesicles. And so these are membrane-bound little just circles, and they contain a signal molecule called a neurotransmitter. And so when calcium enters, that's going to tell that vesicle to start moving towards the end of the cell. And so you can kind of see the membrane of the vesicle fuses with the membrane of the cell so that it can actually release that neurotransmitter, that little red dot. And so my next question for you is, this part where it's releasing all those little red dots, what type of transport is that? So Lou, do you have your poll again? Thank you. So again, the part I'm asking you to look at is there at number 3. Those vesicles are releasing their contents, the neurotransmitter signal, the little red dot. So what type of transport occurs to release that signal?
We'll give about 5 more seconds here. So if you've got a good stab, and the polling is anonymous, students and teachers, so don't be afraid.
All right, good.
I hope that's the right answer, MaryBeth.
Yes, I was just making sure I didn't give it away too soon. So yeah, exocytosis, that material is exiting the cell, and that's a type of active transport. So it does take some energy to rearrange that membrane as the vesicle's membrane is fusing with the cell's membrane. So that actually does take some energy to do that. And so my last question here is this neurotransmitter. So it's a signaling molecule. To send its message to the next cell, it has to get from this cell where it was released to this cell. So how does that molecule, what type of transport allows that little red circle to make it from here down to these little ligand-gated ion channels? So one last poll. And so again,
and MaryBeth,
yeah.
As our participants are choosing their poll, we have a question from a teacher, and that is, do students need to know the terms juxtacrine, paracrine, etc.?
I think those are always good terms to know, but I do not feel like those are ones that they have to memorize. I feel like if it was given, they would explain what it means. So like this would be, these two cells are in close vicinity. So this is a great example of paracrine signaling. But I don't feel like those are terms that they would have to memorize.
And I think that's consistent with what we would expect in other AP courses. Polling looks good. I'm going to give you students about 5 more seconds to make their choices.
All right, good job. Most of you went with diffusion. And so if you look at that picture, remember it's all about high concentration. So there were a lot of those neurotransmitters being released right there. And then they went to a low concentration at that second cell. So good job. I think this is a great picture. Not only does it tie in Unit 2, but Unit 4. And so again, you know, your test is at the end of the year and it's going to tie all these concepts together. So local regulators, these neurotransmitters, allow two cells to talk that are in kind of close vicinity to one another. This does seem to be lagging quite a bit. Okay, so while this one kind of comes into view, I don't know if it's that bad for you guys. Quorum sensing is also a great example of two cells that are in close vicinity being able to talk to one another. And so I find this really fascinating. I have provided a link. Bonnie Bassler, I want to say she does research at Princeton, but anyway, she has a TED Talk, and if you haven't seen this, it really is fascinating. And so she talks about her research. And so in neural studies, bacteria, we know that antibiotics treat bacteria, but through natural selection, they are becoming resistant. So we only have a set number of antibiotics. And so her research is based on this idea, if we can figure out how bacteria talk to one another or how they listen to one another, if we can stop that, that could be our new antibiotics. And so the video is only 18 minutes and it really is fascinating if you have a chance to look at that. And so what's going on here is you have two bacteria cells. And so they're constantly just releasing these green signaling molecules. And so they kind of just diffuse away if nobody's nearby to receive that signal. And so what happens is they don't do anything. And so a lot of times these bacteria, this tiny little prokaryotic cell, when it gets inside of you, it really can't do anything. And so it needs more bacteria cells to actually maybe fluoresce or to actually be, you know, pathogenic within you. And so what happens is as these bacteria cells start to divide, they are going to all start releasing that signal. And so as their density increases, now there's enough bacteria to actually receive that signal and they can say, oh, there's more of me nearby, we can now act together. And so that's how they actually do kind of launch their pathogenicity on you once they're inside. So at this time of year, I usually have students lose this word. And so I would like to ask you guys if you remember. So we'll just go to chat this time. What type of division occurs for bacteria? So how do bacteria replicate themselves? What do we call that type of cell division? So if you'll go to chat real quick, tell me if you remember the name of the cell division that bacteria go through. Yay, you all are making me very happy. So I don't think anybody has said mitosis just yet, so good job. Binary fission or how the bacteria replicate. I'm very happy that you know that. Let me go back one more time. All right. This is probably going to switch on me. Viruses also talk, and so we don't have time to get into the details of how viruses talk to one another, but we are in a pandemic caused by a virus right now. And so I thought some of you might find this interesting. May want to do some research on your own. So there's two articles. So the top one is The Secret Social Lives of Viruses. And the bottom one is also Bonnie Vassler. So that other link that I was telling you about, this is still her research team. And so she's looking into how viruses kind of eavesdrop on our cells and can use that to figure out, should they be replicating and bursting out of our cells, or should they lay low? So these are really two interesting articles if you have the time to look at those. Okay, sorry for the lag time. I'm not sure why it's being so slow today. The last type of signaling that can go on between two cells is long distance communication. And so this usually involves your hormones, and they travel through the blood to get to different parts of your body. So this is one specific example that has come up very often on the AP exam. So before we actually start getting into these details, I'd like us to go to chat again and see if you guys remember the correct sequence of the 3 steps in a signal transduction pathway. So try to put the 3 steps of the signal transduction pathway in the correct sequence in chat. All right, good. So it looks like we're all in agreement again. So reception, transduction, response. And so again, kind of what I would want you to do, so maybe you haven't seen this picture, or even if you have, can you put these numbers to that correct step of the pathway? So, you know, anytime you're given a picture of a cell signaling pathway, I would encourage you, you know, before looking at the question, can you identify reception?
Hello, this is Sue.
Can you identify transduction, and can you identify the response? So take a second, go back to chat, and then just put kind of like reception, and then which numbers in this diagram is where reception is occurring. Same thing for transduction, same thing for response. And so it can be number or numbers for that particular step of the pathway.
And while they're doing that, MaryBeth, if I can ask a question,
Sure.
are those disulfide bridges underneath the arrow with the number 1?
Yes, I do believe that is true.
I'm of
I
course
even paid attention.
as a chemist, and noting that the role of intermolecular forces is going to be pretty crucial here. My second question is, what are those red P's that arrow number 2 is pointing to? Are those phosphates?
Yes.
Wow, I'm 2 for 2 right now.
No,
Okay.
you're good.
Well, that's all I have for now.
Let me see what they're saying. All right, so it looks like we are in agreement again. So hopefully you guys, I hit the wrong button. This is not cooperating with me today.
Mary Beth, I guess sometimes it looks like you've got a web page open that's slowing down your computer. Is that possible? It looks like it might be slowing things down.
I think the only thing, because I'm just presenting my presentation is the only thing. So
Okay, I just thought.
yeah, no, I don't know why it's so
It's
slow.
working fine. I mean, it's slow, but it's working.
All right, so to go back to this picture, if you didn't study insulin, insulin is that hormone that's released when your blood sugar levels are high, and it's going to send the signal to your cells to take in that excess glucose to lower your blood glucose levels. And so reception is occurring here. So you have your protein receptor in the membrane. Your signaling molecule is insulin, that's our ligand, and then steps 2, 3, and 4 are that transduction, so kind of that connection of reception to response. So 5 is our response. The thing that I want to point out that you might see in a diagram, these multiple arrows mean multiple steps. And so more than likely in this picture, that's a phosphorylation cascade. We're going to go over that today. And so then I kind of just want to point out, we have another vesicle here, just like we saw in that previous picture, with a GLUT4 containing vesicle. So that's a glucose transporter protein. And so the only way to get glucose into the cell is to have this transport protein embedded in the membrane. So remember, glucose is a really large molecule. Large or charged particles need a transport protein to actually be able to enter the cell. All right, so this is a very generic picture of a signal transduction pathway, but it gives us an opportunity to kind of talk about those really basic components. And a lot of this stuff we did discuss in our first session if you were with me then. And so again, you're going to probably see a more complicated pathway than this on your AP test, but still take that time to kind of just orient yourself and figure out where you are and what, what's some information that they're giving you some clues about. And so when I look at this, I have a plasma membrane, so I have that phospholipid bilayer. My receptor is found there. And so your receptor is a protein. So think about, we really stress that structure determines function. And so this receptor has a very specific shape. So this is that binding site that allows it to be able to bind to this very specific signaling molecule and respond appropriately. And so receptors and ligands, their interaction is very much like an enzyme and a substrate. And so if you remember back, enzymes, when the substrate binds, the enzyme kind of hugs around the substrate. And that's the same thing that's going to happen here. So the receptor binds to the ligand, and the receptor protein is going to slightly kind of hug around that and change its shape. And when this changes shape, that's the signal for step 2 to occur, the transduction step. So I do want to stress that. Kind of think about all those things that we discussed earlier. So mutations to your DNA could affect the shape of a protein here at the binding site. Remember, a truly functioning protein is made of 2 or more polypeptide chains. So maybe a mutation occurs the shape over here on the intracellular side. And so if that shape's different, that may not trigger step number 2. Environmental changes can affect the shapes of your receptor protein. So pH, increase in temperature, increase in salinity could also affect this functioning appropriately. So anyway, to go back, once this hugs around the ligand and changes shape, that starts transduction. And so the thing that I like about this picture is the 3 molecules that they're showing that's part of the transduction step. Those are usually going to be proteins as well. And they're showing that they're all different shapes. And so that particular shape is going to help facilitate the next step, which facilitates the next step, and so forth. And so again, it's all about that protein having a particular shape and allowing it to do a particular job. And so if, number two, if that protein lost its shape, or maybe it was a different protein inserted there, we either wouldn't get this response or we would get a completely different response.
May I interrupt for just a moment, MaryBeth?
Yes.
So I'm not a biologist, and you know that, and none of the students probably know that. But every time I see a picture like this, there are always three, the transduction seems to involve three separate steps. And I've got a couple of questions. Is it always three, or is that just usually? And then why isn't that just one step? Is there some evolutionary advantage to having, amplification over three steps? So I'll let you, address those if you don't mind.
I don't think it's necessarily three. I think that's probably just a generic. I'm not really sure. I don't know if it just helps emphasize that it's multiple steps or, multiple components to get to that final response. So I don't know why three is the magic number, but it definitely can be more. And I think you'll see that in a couple of slides. The other question is, or the other part, transduction is a chance for the cell to kind of fine tune and make sure that it does have the appropriate and specific response, but it does allow the signal to be amplified. So just one little signaling molecule over here, so that's a very slight change in the environment, can allow the response to be to produce millions of molecules to allow that cell to respond appropriately. So it's kind of, slight changes in the environment still allow the cell to respond appropriately to that change.
Terrific. Thank you so much. And that's all transduction. Those three arrows are all, we call them all
Yes.
transduction.
Yeah. The last thing that I would want to stress in this particular picture is where is the response happening? And so that's why in the previous slide, I asked you guys, could you identify these three steps? I think that's really important because like I said, you're probably not going to have a simple picture like this on test day, but if you can identify where these three steps are occurring, it's going to help you think through how to answer their questions. And so the response here is happening in the cytoplasm. And so for you, if they ask you, what is the response going to be? It's going to be either activating or inactivating a molecule that already exists in the cytoplasm. And usually it is a protein. So the response in this particular cell would be to activate a protein. A lot of times I think we stress the activation, but I do want to point out that sometimes it is just as important to shut down a pathway. So for your cells to respond to whatever environmental conditions are going on, sometimes it is just as important to say, hey, stop what you're doing for a minute and let's figure out what's going on. All right, so again, I feel like, they're going to show you a picture of a cell signaling pathway and they may or may not give you all the information, but based on what we've discussed and gone over, that's going to help you answer the question. And so I want to start with this on the left. And so again, I'm going to orient myself. What am I looking at? So this is my cell membrane, and I've got a protein receptor embedded in it. And so in this picture, they are telling you that this is a water-soluble hormone. So I want you guys to go to chat again. And back in that first session, so if you were with me for the first session, you had a review. Otherwise, see if you guys remember. What are two other names that mean the same thing as water-soluble? I think I got it on that last one. I clicked off of the chat. So hydrophilic, most of you had that one. The other word that you might see, or to help you kind of think through the question, might be polar. And so what I want you to get from this picture is they may not say this is a water-soluble molecule, but if it's docking to a receptor that's found in the membrane, in your head, you should be like, okay, this molecule is polar, it's hydrophilic because it's hanging out here in this watery external environment of the cell. And so here's one other thing I want you to think about. So why is this signal not able to cross the membrane? So we talked about the phospholipid bilayer. That's phospholipids belong to our lipid category. And so we talked about properties of that phospholipid that allowed it to be a good component of the cell membrane. So can you guys tell me why this polar molecule can't cross the membrane? So I'll ask you to go back to chat and see if you can give me a quick answer for that. All right, so a lot of you are going with charge. And so the only thing that I would go back. The only thing that I would want to stress here is, yes, this does have a charge. And remember, the fatty acid tails that make up your phospholipids, those are nonpolar. And so nonpolar and polar don't mix. And so that's how the phospholipids are really selective in what does get across the membrane. So again, the other thing, just to kind of stress, the signal docks to the receptor, the signal is not entering the cell. So I'd like to stress that as well. So you are seeing, again, here's your number, Lou, three proteins making up your transduction part of the pathway. And then they're showing you can either have a cytoplasmic response where we're activating or inactivating a molecule that already exists, or this last protein in transduction that gets activated goes into the nucleus and regulates genes. So it's kind of the same idea over here, lipid-soluble hormone. I just kind of want to stress that that signal is diffusing across the membrane. And so my questions are kind of just the opposite. So what are two other names for lipid-soluble? I'll save us a little bit of time here. It's just the opposite. So lipid-soluble is nonpolar and hydrophobic. And so those phospholipids have those fatty acid tails, which are also nonpolar. And so they're alike, and that allows our signal to actually cross the membrane. The receptor is going to be found in the cytoplasm. So when they come together, they kind of create this complex that will then enter the nucleus, and that complex itself will usually act as a transcription factor to also regulate which genes get expressed. So then this picture is just specific examples of those two in the previous slide. So growth factor could be a signal that tells cells that it is okay to go ahead and start dividing through mitosis. And so it's a water-soluble molecule because it's docking out here. And then testosterone is a great example of a lipid-soluble molecule that can diffuse through the membrane and dock to a receptor inside the cell. So I do want to go back to chat here. So looking at these two pictures, what is the response going to be in these two cells? So go to chat and tell me quickly, what is the response going to be in these two cells? Okay, so a lot of you are saying activation. Activation of what? So what's the response going to be? Okay, so I did get the answer. Some of you are stopping early. So if you look at both pictures, this one on the right does give you a little bit more information. And so mRNA is produced in both. And then this one's kind of not zoomed in as close. Remember, so that's transcription. So a lot of you told me transcription is going to occur. After transcription, we also have translation occur, which means we make a new protein. And this is a great connection of what Brittany heard if you joined her for that second session, combining Unit 6 with cell communication pathways. So now that we know that a protein is going to be made, I'd just like to go back and see if you guys can tell me what's the monomer of a protein, what bond holds those monomers together, and there's actually two words. What kind of reaction did we say had to occur to form that bond between the two monomers? So again, this takes us back to our biochemistry. How much do you guys remember? So see if you can answer those three questions in chat. What's the monomer of a protein? What's the bond that holds them together? And then what was the reaction that allowed that bond to form? All right, good job. It looks like you guys are in agreement again. So the monomer is an amino acid. The bond holding them together, and you guys gave me the specific name, so that's really good. So peptide bond, and we had to remove water to form that bond. So good job. All right, the last thing that I kind of just want to show you in case you were seeing this on your test question, those membrane-bound receptors can look three different ways. So there's a G protein receptor. And so that pathway is going to kind of look like this. It's one of the simplest pathways. You're going to get one response, and this one's highly conserved. So G protein pathways are seen in prokaryotes and eukaryotes. And the other thing that I would want to mention is a lot of our medicines that we take affect a G protein pathway. So I think that's a lot of ways that they could, ask you questions about a G protein pathway. The next kind is here in the middle. This is a tyrosine kinase. We're going to talk about kinases, but kinases phosphorylate other molecules. And this is what you were seeing with that insulin pathway we saw earlier. The difference between this pathway and a G protein pathway, the cell can have anywhere from 6 to 10 different responses from this signaling molecule. And then the last one we saw with our neurons when we started, so ligand-gated ion channels. So if you remember back to that, our neurotransmitter docked here, it kind of opens the gate, and then ions can rush through to kind of send the message forward. All right, so to get back to transduction, I do want to stress this. There's a lot of things that ties in our biochemistry that we learned. And so transduction, this goes back to why is there many steps? It does kind of look like we've got 3 kinases, but we have a 4th protein down here that's being activated before we get a cell response. So the thing that I'd really like you to kind of pay attention to is the shape of these molecules. And so I want to start at the top. So our signaling molecule has bound to our membrane-bound receptor. And again, you obviously can't see that, but it would change shape. So we kind of hug around the signal, and then that's going to initiate this relay molecule. And so that relay molecule's job is to take this inactive protein kinase and activate it. And so what I want to stress here is we kind of have it as an oval shape in the inactive form, but when it's activated, it's now circular. So that shape change allows it now to be activated and do a different specific job. So kinases, if you are seeing that, the ASE ending, remember that tells you it's an enzyme. And kinases specifically, their job is to add a phosphate or phosphorylate another molecule. And so kinase 1 is going to phosphorylate kinase 2. How does it do that? ATP is the source of that phosphate group. And so, again, we go from oval inactive shape to circle active shape, and that continues all the way down until we get our cellular response. So again, you've got many steps. It allows the cell to fine-tune, amplify the signal, so that down here, millions of molecules could be produced from that one signaling molecule. The other thing that I want to stress here is all of this can be reversed. So we don't want, the signal to continue and go on and on and on. We want it to shut back down. And so all these activated enzymes can go back to the inactive state. And it's not written out as well, but this little PP here, if you can see that throughout, that stands for protein phosphatase. And so its job is to remove all those phosphates so the active protein can go back to its inactive form. ATP, also recyclable. So once the phosphate's removed, that phosphate can join back with ADP, and we have ATP again. The thing that's great with being multi-step, that's a lot of resources and it's a lot of energy. And so by being able to recycle those materials, the cell, isn't always having to come up with new materials and new resources to be able to respond appropriately. The other thing that I kind of want to point out here, besides being able to recycle and save resources, is when a cell can produce these proteins and kind of just let them hang out until the cell needs to respond, that's saving a lot of time. So all of these are enzymes. For most of the time, enzymes are proteins. And if you think back to that previous picture, we would have to go. So as soon as that signal was received, this cell would have to go through transcription translation to make this protein, transcription translation to make number 2, and so forth. And that's a lot of time that it probably doesn't have to respond quickly enough to that signaling. So kind of by having them already made prior to needing to respond, that saves a lot of time and it can respond much more quickly. All right, I did want to just give you a good example. So epinephrine in the past, and especially with multiple choice questions, there's always been a lot of questions on epinephrine. And so it's a great signaling molecule to give you an example of this phosphorylation cascade. And so epinephrine is also called adrenaline, and you may know it as a fight or flight hormone. So you're in a dangerous situation and you're either going to fight that situation or flee from that situation. And so epinephrine, the pathway that's going to help your cells respond appropriately is a G protein pathway. So it kind of ties in that G protein receptor. The other thing that I want to point out here for this is sometimes we have second messengers. And I kind of think of them as, you kind of have your first message, and then that talks to a couple of proteins. And then it's like the cell needs to get back together to ensure that it does respond just like that original signal intended. And so CAMP and calcium are great examples of second messengers. In case, that could possibly be a question, I do recommend, CAMP and calcium are second messengers. And so in this picture, CAMP is going to talk to protein kinase A, and then you have two arrows. So that's, it's kind of just removing that phosphorylation cascade that we talked about, but multiple steps are occurring before the cell can respond. And then the picture to the right, same thing, we have epinephrine. You're kind of seeing the details of that phosphorylation cascade, and then you're seeing that idea of amplification out to the right. So at every step, there's more and more molecules of that particular molecule that's helping facilitate that step all the way down. So again, epinephrine, it only takes one signaling molecule to say you're in a dangerous situation, and by the end, 100 million glucose molecules can be released to help your cells fight or flee that change. All right, so another reason I love epinephrine, and I think it does come up, I just want to talk to you about the receptor protein itself. And, why do all cells not respond to a signal? So epinephrine, it's a hormone, it's going to travel long distance, all through your body, through the blood. And so how can different cells respond differently to it, or maybe not respond at all? And so starting with the liver cell and the skeletal muscle cell, they both have the same receptor for epinephrine. But they're going to have different responses. And so how do they do that? It all goes back to that transduction pathway. So if there's different proteins in that transduction pathway, that's going to allow for a different response to occur. And so liver cells, they store glycogen. Glycogen is a polysaccharide of glucose molecules. And when epinephrine binds, you're in a dangerous situation. We want this storage molecule to break apart and release those individual glucoses for energy. Your skeletal muscle, those are going to help you fight or flee. And so when epinephrine binds, we want those blood vessels to dilate so good oxygenated blood can get to your skeletal muscle so it can do that. And then the last example, we finally have a different receptor. So that receptor may be different, but it still has a shape that allows epinephrine to bind. And so now that we have a different receptor, this is an intestinal blood vessel. When you're in a dangerous situation, the last thing we want your body thinking about is digestion. So we want those blood vessels to constrict so that all the good oxygenated blood can get to your blood vessels to fight or to flee. Okay. Lou, I think I started my timer later. Are we about at 40 minutes?
Sorry, MaryBeth, we're at about 48 minutes, but we can go a little bit. Go.
Okay. So we'll try a few of these out, and we may just end up talking about, I'll give you time to actually think in your head about it and then kind of discuss the answer. So we'll I'll just play with the time a little bit. But this was a cell signaling pathway. I think it was given about 2 years ago. And I'd like to walk through this question with you and kind of how to think about it and look at it. So my first suggestion, I think, is to actually look at the diagram. What is familiar to you? And again, orient yourself. What are you looking at? And I think that just helps boost your confidence. Obviously, they're going to give you a question about some biology that you have not heard of. So they're going to give you an example of something that you haven't heard of. And the whole goal is to see if you can apply the biology that you did learn to this new situation. And so I think if we just get into the question or the content that they're providing us with, sometimes we're like, oh my gosh, I've never heard of this, and it makes us nervous and our confidence goes down. So I do really encourage you, take that time to just kind of look at the picture, orient yourself, and that way you can find some stuff that you're familiar with and you take a deep breath and know that you can answer it. So always, I would encourage you to read the label of the picture. So this is a cellular response to infection by pathogenic bacteria, so disease-causing bacteria. And so I'm just going to kind of look through. I see cell membrane, and that's it. So there's no cell wall. So I know I'm looking at animal cell. And then I'm just going to go through, and gasdermin may not sound familiar to you, but it is being inserted into the membrane. So it might possibly be a protein. I go over here, interleukin. Again, that may be something you haven't heard of, but look at the arrow. It's showing you that it's going right through the membrane. And so automatically you can think, okay, so it's either really small or it's non-polar or hydrophobic, and that allows it to cross the membrane without any help. And then if I come on down here, caspase, you may not have heard of that before, but it does have that ASE ending. So you should think that's an enzyme, and enzymes are proteins. And then think about all that stuff that we've discussed that's really important about a protein. And so now I'm ready to kind of look at the question, what other details are they going to provide me with to answer? And so some pathogenic bacteria enter cells, replicate, and spread to other cells, causing illness in the host organism. Host cells respond to these infections in a number of ways, one of which involves activating particular enzymatic pathways. So now they are telling us it's enzymatic pathways, so these are protein pathways. Cells normally produce a steady supply of inactive caspase-1 protein in response to intracellular pathogens. So I do intra means within. So that pathogen has already made its way inside the cell, and that is what we're seeing in this diagram. The inactive caspase-1 is cleaved and forms an active caspase. So that's what we're seeing in step 1. So it's inactive here, and then once the cell's infected, that will be cleaved so that it's now activated. Active caspase-1 can cleave to other proteins. So, when we initially looked at this, we didn't know what gasdermin or interleukin was, but now they're telling us that those are protein molecules. So again, everything that we know about proteins applies to these two molecules. When caspase-1 cleaves an inactive interleukin, so step 2, the active portion of interleukin is released from the cell. And so then they tell us what interleukin actually does. It's a signaling molecule that can activate the immune response. So now we know exactly what interleukin does. When caspase-1 cleaves gasdermin, step 3, the N-terminal portions of several gasdermin proteins associate in the cell membrane to form large, non-specific pores. So we were correct. This was a protein embedded in the membrane. They're telling us it's non-specific, so a variety of different molecules could actually enter through this pore. We're going to come back to this diagram. So this is some more information that was provided with this question. All right, so let's go ahead and try for A. Let's go to chat. The maximum you should be giving me is a sentence, if that. And it's asking you to describe the effect of inhibiting step 3 on the formation of pores and on the release of interleukin from the cell. So I'm going to go back to the picture, but your job in chat is to tell me the effect of stopping step 3 on the forming of pores and on the release of interleukin. So again, go to chat. If step 3 was inhibited, so if I put a big X here, what would be the effect on forming this pore And what would be the effect on releasing interleukin from the cell? So step 3 is stopped. Tell me the effect on forming a pore and on interleukin being released. So again, while you guys are kind of thinking, you don't have to repeat questions, save yourself that time. And so you're telling me if step 3 could not occur, how does that affect the pore forming and interleukin being released? All right, so you guys are doing good. So I really like this question. So only step 3 was stopped. So that means that that pore cannot form, but that step has nothing to do with interleukin being released. So good job. If you said that, you got 2 points right off the bat. All right, so for part B, they want you to make a claim about how cleaving the inactive caspase results in an activated caspase. So again, just about a sentence, you're making a claim on how cleaving or cutting the inactive caspase results in it being activated. So I'm going to go back so you can see the picture. So here's our inactive caspase. How does cleaving it? So if you look in this picture, we're just kind of cutting off that little black circle, and now it's active. So it's just kind of the white oval. How does that activate it? How does cleaving? So you're just making a statement of how just cutting that part off activates that molecule. So again, I'll ask you to go to chat. All right, so you guys are doing good. I'm going to go ahead and move on so we can answer a little bit more. But you guys answered that perfectly. So some of you, so there was kind of 2 ways. You could be specific and kind of say this was an inhibitor. So this little black circle kind of inhibited caspase from being active. So that was removed. Or what I would want you to get back to is just that simple biochemistry. We have changed its shape, and so therefore we've changed its function. And so if you said that, you earned yourself a point. Okay, so for the second part of B, they're telling us that a student has now made a claim that pre-infection production of inactive precursors shortens the response time of the cell to a bacterial infection. And they want you to provide one reason to support that student's claim. So again, this would just be a sentence. So a student is claiming that pre-infection production of inactive precursors shortens the response time to this bacterial infection. So I'll go back so you can kind of see that. So the student is saying that by having these inactive proteins already in the cell prior to being infected, that's going to shorten the time it takes your cells to respond. So can you support that claim? So again, I'll have you go to chat if you haven't already, kind of a sentence maximum. All right. So if you're still working on that, a lot of you, I mean, you're giving great answers. So you're telling me that we're not having to wait. The protein's already there. All we have to do is cleave them and they're activated. We're not having to waste time going through that transcription translation process to produce a protein. And so I think there was two ways you could word this one too. You could say the inactive forms were already there. They just have to be cleaved. We're not having to wait to produce the protein, or you could word that we're not having to wait through transcription and translation to produce a protein. That was one point as well. So if you said something along those lines, you earned yourself a point. Okay, so I kind of want to skip C. C uses that other diagram, so I don't want to get off course just yet. I want to stick to the diagram we've been looking at. So D is telling you to describe the most likely effect of gas dermin pore formation on water balance in the cell if it's in a hypotonic environment. So remember that gas dermin pore formation, they told us that it was non-specific. And how would water balance change if this cell found itself in a hypotonic environment? So again, a sentence is all you need to answer this question. Okay, so we've got some opposing views on this. All right, it looks like the majority of you are saying that water would enter the cell, and that's exactly all you had to say. Water will enter the cell. Something that I want to stress, or I try to tell my students, hypotonic, if that's the environment a cell is in, water is going to move in and cause that cell to swell like a big O. And so in hypotonic, water should move into the cell and cause that cell to swell like a big O. So see if that helps you remember hypotonic, if you got them flipped. So if you said that, if you said water is going to enter the cell, you earned yourself a point for D. And then E, you kind of had to understand D correctly, I think, to answer part of E. So it's asking you to explain how gas dermin pore formation and interleukin release contribute to an organism's defense against this bacterial pathogen. So you want to explain how this pore is defending the cell and how interleukin is defending the cell. And so I'll go back to your picture. Maybe, I'm sorry, this has been so slow today. So they're wanting you to tell them, how does this pore help protect the cell, and how does releasing interleukin help protect the cell? And so again, go to chat. One sentence, maybe two here. All right, this is not cooperating. I can't get the chat to pop up again for me.
I think people might be a little shy on this one, Mary Beth. So why don't you go ahead and just explain the what the question is looking for?
Okay. So on this one, you kind of, if you understood D and you said water was going to enter, you have to think about what's going to happen to that cell. And so this cell, if you remember, it doesn't have a cell wall. And so if water is entering the cell, eventually that cell is going to burst or lyse. And that doesn't sound like a good thing for a cell, but if it's infected with the bacteria, that's going to stop the spread of that bacteria. So it does kill this cell, but it does prevent that bacteria from replicating and spreading further. So that's the benefit of the pore. The benefit here for interleukin, it goes back to what they told you here. And so sometimes if you feel like you totally don't know how to answer a question, sometimes they kind of give you the answer in the question stem. So they told us earlier that an interleukin is a signaling molecule that can activate your immune system. And so that's what you'd want to say to get that point, is that interleukin would leave the cell and go talk essentially to other immune cells to come back and help take care of this bacterial infection. So somewhere along those lines, the pore is going to cause the cell to lyse or burst because water has moved in. And the interleukin helps because it has gone out and activated immune cells to fight this pathogen. And so that would be 2 points. So I didn't have access, I couldn't pull the chat. If you said those 2 things, you would have earned 2 points.
Well, thank you, Mary Beth. That's been a little bit more than an hour, and thank you all those who are patient and I think learning some really good stuff here. Do you have any final or, oh, I'll let you do this and then maybe we can wrap this up.
Okay. Yeah. So the only thing that I wanted to point out here is I feel like a lot of you in this class, you feel like you have to be perfect and get all 10 out of 10 points to pass the test. And that's just not the case. And so this question, so this is the results. So this was given about 2 years ago across the world. And the mean score was 2.68. So if you had earned 3 points, so kind of think back about what we discussed. If you earned 3 out of the 10 points, you would be above the mean, and you're really setting yourself up to have a passing score. And so obviously all that's different now that we only have 2 questions, and I can't really tell you more because I don't know how all the statistics work. But as long as you're beating the mean, for most biology questions, that's getting 4 out of 10 points. So you don't have to get every single one. Answer what you know. And so I think that's going to be easier to kind of fill in. Since you're typing this year, you can kind of go back and add when you realize you remember something. But answer what you know. Don't leave it all blank because you don't know one part. So go through. There's always easy, you know, couple points that you can get that doesn't always stick out to you initially. So this is my last session. If you have questions, marybeth.cook@harrison.kyschools.us is my email. I'm happy to answer those. But again, I do wish you the best of luck on your test. So take care.
And thank you very much. And thank you, students. And again, this will be archived on KYAP 2020. And we will hopefully look forward to seeing you for the mock exam practice and exam preparation sessions. Best of luck and take care. Bye-bye now. I'm going to sign off.